Welcome to the deep dive. Today, we're tackling something really fascinating, actually, a bit of a paradox. It's about oxygen. You know, the stuff we absolutely need to live, every breath, every cell needs it. But here's the twist, that same oxygen can sometimes, well, turn destructive inside our own bodies. How does that work? So our mission today is to really get to grips with this. We're diving into oxidative stress and the body's amazing antioxidant defenses. We want to equip you, our future clinicians, with a solid understanding of this constant molecular battle, make these concepts stick. Because knowing this balance or imbalance is key to understanding so many diseases. Ever wonder how the body keeps things stable at that tiny microscopic level? And what happens when it gets knocked off balance? Let's jump in.
It's a great place to start. It's truly fundamental, this constant tug of war between oxidants and antioxidants. It's happening all the time in our cells.
Right, and understanding that battle means going back to basics, doesn't it? Let's talk redox reactions. Can you give it a quick refresher?
Yeah, absolutely. Good starting point. So a redox reaction, it's basically where oxidation and reduction happen together. You can't have one without the other.
Okay.
Think of it like electrons being passed around. Oxidation is the loss of electrons. Reduction is the gain. We often use the acronym OIL-RIG to remember that.
Oil or rig. Got it. Oxidation is loss, reduction is gain.
Exactly. So an oxidizing agent takes electrons, it gets reduced. A reducing agent gives electrons, it gets oxidized. This electron exchange is driving, well, countless processes in the body. Energy, detox, you name it.
So that's the normal balanced exchange. But what happens when things get messy? That's where free radicals come in.
Precisely. Normally, electrons like to hang out in pairs, like buddies.
Okay.
Stable. But a free radical. That's an atom or molecule with one or more unpaired electrons. It's missing a partner.
Like a lone wolf.
That's a good way to think of it. It's desperate to find another electron, which makes it incredibly reactive, chemically unstable. We often show that unpaired electron with a little dot, like oh guy.
And these reactive guys, these electron thieves, they're not picky, are they? They'll just snatch an electron from whatever's nearby.
That's the problem. They grab an electron from a stable molecule, damaging it. And often that molecule itself then becomes a free radical.
Oh, so it starts a chain reaction, like cellular chaos.
Exactly, a cascade of damage.
Yeah.
And when the production of these reactive species, these free radicals, gets too high, or the body's defenses can't keep up, that is what we call oxidative stress.
Okay, oxidative stress. So it's an imbalance.
That's fundamentally an imbalance.
Oh.
The generation of ROS, that's reactive oxygen species, and RNS, reactive nitrogen species, it just exceeds the body's capacity to neutralize them or repair the damage they cause.
So think of it like a fire inside the cell. If the sparks, ROS and RNS production are flying faster than the firefighters, our antioxidants can douse them.
That's a perfect analogy. You get widespread damage, proteins get hit, lipids and membranes, even our DNA. And this damage is a really significant factor in, well, a huge list of diseases.
Okay, that makes sense. So you mentioned ROS and RNS, these cellular arsonists. Let's dig into them. What are they exactly and where do they come from?
Right, so the main culprits, especially for ROS, reactive oxygen species, are actually our own mitochondria, the power plants of the cell.
From generating energy.
Yeah. They use the electron transport chain, the ETC, to make ATP. It's mostly efficient, but it's not perfect. Some electrons inevitably leak out, maybe around 0.1 to 2%.
And that leak is the start of the trouble. An electron escapes, finds an oxygen molecule.
Exactly. When an electron adds to molecular oxygen, O2, you get superoxide. That's O2, often the first ROS formed, the initial spark.
Okay, superoxide. What happens next?
Well, superoxide can then be reduced, gain another electron essentially, to form hydrogen peroxide, H2O2. Now, hydrogen peroxide itself isn't a free radical, no unpaired electron, but don't underestimate it. It's still a potent oxidizing agent, and it can diffuse quite easily through membranes.
And it can cause more trouble downstream.
Big trouble. Especially if it bumps into transition metals like iron or copper, which are often around in biological systems. Through something called the Fenton reaction, H2O2 plus iron generates the hydroxyl radical.
OH.
Hydroxyl radical, that sounds bad.
It is bad. It's often called the wrecking ball of the cell. Extremely reactive. It will react with pretty much the first thing it encounters. DNA, proteins, lipids, causes major damage.
Wow, okay, so that's the oxygen side, ROS. But you also mentioned RNS reactive nitrogen species. How does nitrogen get involved?
Well, remember superoxide, O2O. It doesn't just react with oxygen-related things. It can also react with nitric oxide, NO2.
Nitric oxide, isn't that important for signaling, like blood vessel dilation?
Exactly. O-No is a crucial signaling molecule. But when it reacts with superoxide, bam, you get peroxynitrite, ONO.
Peroxynitrite, is that a radical?
No, not a radical itself, but it's a very strong oxidant and nitrating agent. It basically shuts down nitric oxide's useful functions and causes its own brand of cellular damage, like nitric oxide's evil twin, really. Okay, so these reactive species are generated inside, mainly mitochondria. But what about outside sources? Are we getting hit from the environment too?
Are absolutely. We can group the sources.
Mm.
Endogenous, meaning
Yeah.
internal, that's the ETC leak we talked about, but also some enzymes generate them as byproducts, things like peroxidases, nitric oxide syntheses, and those enzymes, NADPH oxidases, especially in immune cells, xanthine oxidase.
So normal cellular processes can contribute.
Yes, often related to metabolism or inflammation, but then you have the exogenous sources from outside, things like radiation UV light from the sun, X-rays, environmental pollutants in the air we breathe.
What about things we ingest?
Certain drugs, definitely. Some antimalarials like primaquine, even high doses of paracetamol can cause oxidative stress in the liver, and toxins like the herbicide paraquat or even radioactive stuff like polonium-210. So yeah, we're constantly exposed inside and out.
Okay, so we've met the aggressors, ROS and RNS, and we know where they come from. Now, the damage report. What exactly gets hit inside the cell when these reactive species run wild?
They're not picky, but they have preferred targets. Major biomolecules. Let's start with DNA. ROS can directly attack the bases in DNA. Guanine is particularly susceptible.
What happens when a base gets attacked?
It can be modified, chemically changed. This can lead to mispairing during DNA replication. So G might pair with A instead of C, for instance. That's a mutation. ROS can also cause breaks in the sugar phosphate backbone of DNA, single or even double-strand breaks.
Is there a way to measure this damage, like a marker?
Yes, one well-known marker is 8-oxo-2-deoxyguanosine, or 8-oxo-DG. It's basically an oxidized form of guanine. Finding higher levels of 8-oxo-DG tells us there's been significant oxidative DNA damage.
And you mentioned mitochondria earlier. Is their DNA affected too?
Hugely affected. Mitochondrial DNA, mtDNA, is particularly vulnerable. Think about it. It sits right next door to the main ROS factory, the ETC. Plus, unlike nuclear DNA, it doesn't have those protective histone proteins wrapped around it.
So it's kind of exposed.
Very exposed. And damage to mtDNA is heavily implicated in aging and many diseases. If the damage isn't repaired, it can lead to mutations, mitochondrial dysfunction, and contribute to things like cancer development.
Okay, DNA damage. What else gets hit?
Proteins. Proteins are major targets. ROS can modify the amino acid side chains, oxidizing them, adding carbonyl groups, cross-linking proteins together. They can even break the protein backbone itself, leading to fragmentation.
And what does that do to the protein?
It usually destroys its function or alters it in harmful ways. Misfolded proteins can clump together, forming aggregates. A key target is the amino acid cysteine, which has a sulfur group. ROS can cause inappropriate disulfide bonds to form between cysteine residues, leading to misfolding or cross-linking.
Is there a clinical example of this protein damage we might see?
Absolutely. A classic one is Heinz bodies and red blood cells. Remember those?
Vaguely.
Yeah.
Dark spots.
Exactly. They're these dark staining clumps you can see under a microscope. They are basically precipitates of damaged cross-linked hemoglobin, the protein that carries oxygen. Oxidative stress causes the hemoglobin to denature and aggregate.
And what happens to red blood cells with Heinz bodies?
These damaged proteins stick to the inside of the red cell membrane. This makes the cell less flexible, more rigid. When it tries to squeeze through narrow capillaries, especially in the spleen, it gets trapped and destroyed.
Leading to anemia.
Precisely. Hemolytic anemia. It's a direct consequence of oxidative protein damage.
Okay, so DNA, proteins. What's the third major target?
Lipids, especially the polyunsaturated fatty acids found in cell membranes. The reaction of ROS with these lipids is called lipid peroxidation.
Peroxidation sounds like a chain reaction again.
It is. One radical attacks a lipid, creating a lipid radical, which reacts with oxygen to form a peroxyl radical, which then attacks another lipid. It propagates. This cascade severely damages cell membranes.
How does it damage them?
It disrupts the structure. Remember, membranes need that fluid hydrophobic barrier. Lipid peroxidation introduces polar groups, breaks fatty acid chains, decreases fluidity, and ultimately compromises the membrane's integrity. It basically makes the membrane leaky and dysfunctional.
And the consequences?
Huge. Membrane damage affects everything from ion transport to cell signaling. It's thought to be a key early step in atherosclerosis, the buildup of plaques in arteries, contributing to cardiovascular disease.
Wow. So DNA mutations, non-functional proteins, leaky membranes, it's clear this is serious stuff. Our cells are constantly under fire. But thankfully, we fight back, right? Tell us about the body's defense system, the antioxidants.
Yes, absolutely. We have a really sophisticated multi-layered defense system. Think of them as the firefighters and the cleanup crew. We can broadly divide them into enzymatic and non-enzymatic defenses.
Okay, let's start with the enzymes, the elite squad.
Right. Three key players here. First, SOD, superoxide dismutase. This is often the first line of defense. Its job is to take that initial superoxide radical, O2, and quickly convert it into hydrogen peroxide, H2O2, and molecular oxygen, O2.
So it deals with the first spark.
Exactly. Gets rid of the highly reactive superoxide, but now you have hydrogen peroxide. So second, you need CCAT catalase. This enzyme is mainly found in peroxisomes and is super efficient at breaking down hydrogen peroxide into harmless water and oxygen. It's a major H2O2 scavenger.
Okay, SOD deals with superoxide, CCAT deals with hydrogen peroxide. What's the third one?
The third major one is GPX, glutathione peroxidase. This enzyme also tackles hydrogen peroxide, reducing it to water. But crucially, it needs a helper molecule called glutathione to do this. GPX can also neutralize lipid peroxides, those damaging products of lipid peroxidation we just talked about, protecting cell membranes.
Glutathione. That sounds important. Is that part of the non-enzymatic defense then?
Yes, glutathione is a cornerstone of the non-enzymatic system. It's a small molecule, a tripeptide, that our cells make. We usually talk about its reduced form, GSH.
GSH, what does it do?
GSH acts like a rechargeable battery for the cell. It readily donates an electron, specifically from its thiol group, a GSH, to neutralize ROS directly or indirectly via GPX. In doing so, GSH itself becomes oxidized.
And what's the oxidized form called?
Two oxidized GSH molecules link up via a disulfide bond to form GSSG, oxidized glutathione.
So the battery's been used. Can it be recharged?
Yes. And this is critical. To be useful again, GSSG needs to be reduced back to 2 molecules of GSH. This reaction is carried out by another enzyme, glutathione reductase, or GSR.
Okay, so GSR recharges the glutathione battery. Does GSR need anything to work?
Ah, yes. And here's a crucial link. Glutathione reductase requires NADPH as a cofactor, an electron donor, to reduce GSSG back to GSH.
NADPH. Okay, this sounds important. Where does the cell get NADPH?
Primarily from a metabolic pathway called the pentose phosphate pathway, or PPP. This pathway runs parallel to glycolysis. Its main job isn't ATP production, but generating NADPH and precursor sugars.
So let's anchor this thought for everyone listening. The pentose phosphate pathway makes NADPH. NADPH is essential for glutathione reductase to recharge glutathione, GSH. And GSH is vital for neutralizing ROS via glutathione peroxidase. It's all connected. No NADPH, no GSH regeneration.
And your antioxidant defenses are severely weakened.
Exactly.
It's a critical metabolic link.
Okay, beyond glutathione, what other non-enzymatic defenders do we have? Do we get help from our diet?
We absolutely do. Several vitamins are key antioxidants. Vitamin E, specifically the form called alpha-tocopherol, is lipid-soluble. It lives in cell membranes.
Protecting them from lipid peroxidation, like a bouncer at the membrane club.
Yeah, that's a good way to put it. It intercepts radicals within the membrane, breaking that chain reaction. Then there's vitamin C, ascorbic acid. It's water-soluble.
What does vitamin C do?
One of its really important roles is actually to regenerate vitamin E after it's been oxidized. Vitamin E sacrifices itself. Vitamin C brings it back to its active, reduced form.
So vitamin C is like vitamin E's personal assistant, keeping it ready to work.
Kinda, yeah. They work together synergistically. We also get antioxidant benefits from other dietary components, carotenoids like beta-carotene, flavonoids found in fruits and vegetables, trace minerals like selenium and zinc, which are cofactors for antioxidant enzymes. Even uric acid and the hormone melatonin have antioxidant properties.
Wow, quite a team. Now, you mentioned NADPH oxidase earlier in the context of immune cells. That sounds like it makes ROS, not defends against them.
Yes, the respiratory burst. This is a fascinating specialized case where the body intentionally produces massive amounts of ROS. It happens in phagocytic immune cells, like neutrophils and monocytes, when they encounter pathogens like bacteria.
So the immune system uses ROS as weapons, like bringing out the heavy artillery.
Exactly. The enzyme NADPH oxidase gets activated and generates a huge burst of superoxide. This is rapidly converted to hydrogen peroxide, H2O2. Then another enzyme specific to these cells, myeloperoxidase, MPO, uses H2O2 and chloride ions to produce hypochlorous acid, HOCl.
HOCl, isn't that basically bleach?
It is. It's a very potent antimicrobial agent. The immune cells essentially unleash bleach onto the bacteria they've engulfed. helping to kill them. It's a powerful, albeit potentially damaging if uncontrolled, defense mechanism.
So what happens if this weapon system fails? Is there a clinical condition related to that?
Yes, there is. A genetic defect in any of the subunits of the NADPH oxidase enzyme complex causes chronic granulomatous disease, or CGD.
What happens in CGD?
Patients with CGD, their phagocytes can engulf bacteria, but they can't produce that respiratory burst. They can't make the superoxide and downstream ROS needed to kill certain types of bacteria and fungi effectively.
So they get recurrent infections.
Severe recurrent infections, often with specific organisms that are catalyst positive, meaning the bacteria can break down any H2O2 the patient does manage to make. They also tend to form granulomas, which are collections of immune cells trying to wall off infections they can't clear.
Yeah.
It really highlights how important this ROS-generating system is for immunity.
That's incredible. Okay, so we've seen the attackers, the damage, the defenders, even how we use ROS offensively. Let's tie this all together clinically. How does this microscopic battle translate into diseases we see in patients? What does this mean for a first-year med student trying to understand pathology?
Well, as we touched on, oxidative stress is implicated across the board in chronic diseases. Think cardiovascular disease, ROS, contribute to endothelial dysfunction, and the modification of LDL cholesterol, key steps in forming atherosclerotic plaques.
Cancer too.
Yes. DNA damage from ROS can lead to mutations that drive cancer initiation and progression. Neurodegenerative diseases like Alzheimer's and Parkinson's show evidence of significant oxidative damage in the brain. Also, inflammatory conditions like rheumatoid arthritis, type 1 diabetes, eye conditions like cataracts, even the aging process itself is thought to involve accumulated oxidative damage.
So it's rarely the single cause, but a major player?
Generally, yes. It's often intertwined with inflammation, mitochondrial dysfunction, and other cellular stresses. But it's a common thread and a potential therapeutic target in many conditions.
Okay, let's circle back to something you mentioned earlier, the NADPH link. Here's a question to make us think. Why would a specific enzyme deficiency make someone sick just from eating, say, fava beans?
You're leading us to glucose-6-phosphate dehydrogenase deficiency, or G6-PDH deficiency, a classic example.
Right. Remind us, G6-PDH is the first enzyme in.
The pentose phosphate pathway, the pathway that generates the bulk of NADPH, especially in red blood cells.
Okay, so G6-PDH deficient means less NADPH, and we established that less NADPH means
Less ability to regenerate reduced glutathione, GSH, using glutathione reductase.
right. The rechargeable battery runs low.
Exactly. And red blood cells are particularly vulnerable because they carry oxygen constantly, so they face a high oxidative load. They also lack a nucleus, so they can't make new enzymes or repair proteins easily.
So under normal conditions, maybe they cope, but add an extra stressor.
Like certain drugs, antimalarials, some antibiotics or infections, or indeed compounds found in fava beans, these things increase oxidative stress. In someone with G6-PDH deficiency, their already limited antioxidant capacity due to low NAD, BPH, and GSH gets overwhelmed.
Leading to
Damage. Hemoglobin gets oxidized, forms those Heinz bodies we talked about. The cell membrane gets damaged. The red blood cells get destroyed prematurely in the spleen. Result, hemolytic anemia.
wow, all from that one enzyme link back to NADPH and glutathione. It's an X-linked condition, right? More common in certain populations.
Yes, X-linked recessive. So mostly affects males. It's common in people of African, Mediterranean, and Middle Eastern descent. Interestingly, like sickle cell trait, being a carrier is thought to offer some protection against malaria, which might explain its prevalence in those regions.
Fascinating connection. Are there other genetic conditions where this NADPH depletion is a problem?
Another example, perhaps less common but illustrative, is galactosemia. This involves defects in enzymes needed to metabolize galactose, the sugar found in milk.
How does that relate to oxidative stress?
Well, if galactose isn't metabolized properly, it gets shunted into an alternative pathway where an enzyme called aldose reductase converts it to galactitol.
Okay.
The problem is aldose reductase uses NADPH as a cofactor. So high activity of this enzyme trying to deal with the excess galactose actually consumes NADPH.
So it depletes the NADPH needed for glutathione reductase again. Different starting point. Same downstream problem for antioxidant defense.
Precisely. And this contributes significantly to some symptoms of galactosemia, particularly the formation of cataracts. The lens of the eye is very sensitive to oxidative stress. The depletion of NADPH compromises its defenses, leading to damage and aggregation of crystalline proteins, causing the lens to cloud up.
It really hammers home how central that NADPH glutathione system is. Okay, one last question to reflect on. We started by saying oxygen is vital. But why can too much oxygen be harmful? We sometimes give patients high concentrations of oxygen, don't we?
We do, when necessary. But it's a double-edged sword. The condition is called hyperoxia. Breathing high concentrations of oxygen for prolonged periods can actually overwhelm the body's antioxidant defenses.
Oh, does it just generate more ROS?
Essentially, yes. More oxygen available potentially means more electrons might leak from the ETC and form superoxide. It increases the overall oxidative burden. This is particularly concerned for the lungs, obviously, as they're directly exposed. It can damage the delicate lining of the bronchioles and alveoli.
Can it affect other areas?
Yes. For example, in premature infants given high oxygen, it can damage the developing retina, a condition called retinopathy of prematurity. It can also pose risks for adults on long-term ventilation. It reinforces the idea that homeostasis is key. Too little oxygen is bad, but too much can also be toxic. More is not always better.
That's a powerful takeaway. So wrapping up, we've gone from basic electron swaps and redox reactions, met the free radical lone wolves, seen the damage they cause to DNA, proteins, and lipids, explored our amazing antioxidant firefighters like SOD, catalase, and the crucial glutathione system linked to NADPH.
And we've seen how breakdowns in this system, whether genetic like G6PD-H deficiency or galactosemia, or due to external factors like drugs or excess oxygen, lead to real clinical diseases.
Understanding this balance, this constant molecular tug of war, it isn't just abstract biochemistry. It underpins so much of the pathology you'll encounter as clinicians.
Absolutely. It connects metabolism, genetics, environment, and disease in fundamental ways.
So as you continue your studies, think about this internal balance. Consider how everyday things, diet, lifestyle, environmental exposures might be subtly tipping the scales within our cells. How can knowing this empower you, not just to treat disease later, but maybe to think about prevention and health maintenance for your patients and even for yourselves? Keep asking those questions, keep digging deeper.